Okay, so a quick review of what we already studied. We've looked at passive transport. Now, if you remember passive transport, this was molecules moving from an area of high concentration to an area of low concentration. And the key here part here, the passive part, was that no ATP or energy was required. Now we studied three types. The first type we talked about diffusion, which is simply the movement of molecules from high to low with no energy being required. The second one we looked at was osmosis. Now in osmosis, we talked about tonicity. We talked about solutions that were isotonic. We also talked about solutions that were hypertonic. And the last one we talked about were solutions that were hypotonic. Now remember, hypo, the hippo gets fat, the cell gets fat. Whereas in hypertonic, hyper people get skinny. So that cell in a hypertonic solution would get smaller, would get skinny. And in an isotonic solution, that cell would stay the same. And the last type of passive transport that we talked about was facilitated transport or facilitated diffusion. And so now what we need to discuss is in a little bit more detail, as we didn't in the last podcast, is that active transport. So in active transport, this is the opposite. We're moving molecules from an area of low concentration to an area where they are already highly concentrated. Because we are moving against the concentration gradient, these do require ATP. And typically we're looking at where molecules are moving to an area where they're already concentrated and the protein usually has to move. And that's where the ATP or energy comes in. So this podcast will be on active transport. And as you can see in this animation here, there's the phospholipid bilayer that we've already dissected. And here is a protein channel. This protein channel will be like the gated ones we talked about, in that it has to open in order to allow this substance here to move through it. And that's where the energy comes in. In order for things to move, we typically need ATP. Okay, so let's start off with defining active transport, and this is the movement of polar, large, and charged molecules, and they're going to move against their concentration gradient. And what this means here is just simply that low concentration to that high concentration that exists. That's against the concentration gradient. So again, this requires ATP. So as you can see in this animation here, what we're looking at is here's a cell membrane and here is a protein channel or a protein carrier that is going to move these sodium ions and these potassium ions here. And so let's reset to the beginning. Here we have ATP and sodium within the cell. Now that sodium, when in the presence of ATP, it's going to phosphorylize, and this means that that high energy bond of that lost phosphate group is going to be removed, and then when it breaks that bond, a whole bunch of energy is released. And in that breaking of that bond, we end up with adenosine diphosphate, ADP, in order to get that sodium to move to the outside of the cell. Now, same is true in the reverse. When we get those three sodium out of the cell, the potassium molecules, these blue ones here, again, they will fit into this protein structure, a very specific shape, and will utilize that ATP in order to allow the potassium to move inside the cell. This is very typical, as we're going to discuss in our first type of protein pump, the sodium-potassium pump. This occurs quite a bit in the nervous system. And so here's some examples of things that will move by active transport. There's that sodium and potassium as shown in the animation, as well as some sugars, amino acids, and some nucleotides will also move by active transport. And so here's the three type of active transport that we are going to look at. Sodium-potassium pump. It is a protein pump. Another process is called endocytosis, in which can be broken down into two different types, phagocytosis as well as pinocytosis. And the last one is EXOCYTOSIS. So that sodium-potassium pump is what we've just described in that last animation. Here we have sodium which attaches to the side of the protein pump. It utilizes ATP. That ATP will be phosphorylized, which means that last phosphate group, which has the high energy bond, that bond is broken. And when it does so, it produces that ADP. And therefore, when that protein pump allows those three sodium to bond here, the protein will change shape in the presence of that ATP, which allows the sodium to be released to the outside of the cell. This shape now allows the potassium molecules to bind, or the potassium ions, I should say, to bind to the protein pump, which therefore changes its shape again back, and therefore we get the potassium to move to the inside of the cell. And so this then resets and continues over and over again. Again, this occurs in the nervous system, and that's where we're mostly going to discuss this in biology. And so why are we discussing active transport in Biology 12? Well, as I've already said, in the nervous system, here's a nervous system cell called a neuron. In order for that information to travel along this cell so that we can transport the information from the brain to another part of your body, whether it be for a muscle contraction or release of a hormone, we need those sodium-potassium pumps to exist and actively transport sodium and potassium all along the cell membrane of this neuron, the cell within the nervous system. Another place is within the thyroid. So this is a diagram of a thyroid that we're showing you here. And the thyroid relies on active transport of iodine. And so iodine will be actively transported. And in the presence of that iodine, our thyroid will release many hormones. Now we can have something called hypothyroidism, in which can lead to goiter. And this means that your thyroid becomes enlarged because of an iodine deficiency. All right, second type of active transport is called endocytosis. Now this term endo, we'll come back to it again later on in Biology 12. This means in. So what we're talking about here is the invagination of molecules that will form a vesicle in order for these molecules to get into the cell. So these molecules are moving into the cell. And again, because this is active transport, it does require energy. So moving in. Okay, so here's a diagram also showing you that endocytosis. So this light yellow line along here, this is the cell membrane. So this is the outside of the cell, as I've written here, and this is the inside of the cell. So as molecules are moving close to the cell, they may go through an invagination process in order to form a vesicle and get those into the cell. So this is endocytosis. There are two types of endocytosis. One is called pinocytosis, which means small drinking. I always think that term pino, it means small, and it sounds small to me. So small molecules that are ingested and vesicles form. So here's the example in this animation off to the right here. Here's the small molecules that exist outside the cell membrane, solutes, and when they come into the cell, they can't just transfer across the cell membrane, and so therefore they must enter in through forming a vesicle. The second type of endocytosis is called phagocytosis. So this is cell eating because these are large particles that are ingested and formed into a vesicle. So this is an example where a white blood cell will eat a bacteria, and therefore, as you can see here, it will actually fuse with a lysosome. That lysosome contains digestive enzymes, and so therefore that lysosome will digest that bacteria within the vesicle. Now I've shown you these two diagrams here because I want to remind you that this cell membrane that we see here, I'll just give it a color for you. So this cell membrane that we see here, it's a phospholipid bilayer. So this is a reminder that whether it is pinocytosis, these small molecules entering in, or whether it's phagocytosis with larger molecules entering in, these vesicles will still be composed of the cell membrane because they formed a vesicle from the cell membrane. And this last process here is exocytosis. And so exo means out. It's the opposite, the reverse of endocytosis. And so this is where we look at the cell releasing contents of a vesicle to the outside of the cell membrane. So in this diagram here, this would be the inside of the cell, this would be the outside of the cell, and here's this phospholipid bilayer that we see. So the vesicle that is within the cell is moving towards, and yes, it would be moving along. Do you remember which part of the cytoskeleton it was? Right, so hopefully you said microfilaments, that these vesicles will be actually being walked along a microfilament, part of the cytoskeleton, to get to the outside of the cell, to fuse with the plasma membrane and release its contents to the outside. So this is mostly utilized when we're talking about the endoplasmic reticulum with its ribosomes attached have produced a protein. That protein was moved in a transport vesicle to the Golgi apparatus or Golgi body. The Golgi then packages up, might modify those proteins and releases it in a vesicle. This is the vesicle that is released from that Golgi apparatus that will move along the microfilament and meet up with the plasma membrane to release those proteins to the outside of the cell. So here's my question to you. There needs to be a balance between endocytosis and exocytosis. And I want you to think for a minute before you take this last question of the quiz and try to figure out why there needs to be a balance. I'm going to give you a hint. It has something to do with the cell membrane. And that's the end of our Active Transport podcast. Do not forget that there is always resources available for you on the Biology 12 resource page. So right now we are in Unit 3 looking at cells. And if you scroll down, you are going to see that there are many animations typically available to you in order to look into these topics in a little bit more detail. Specifically looking at right now, the endocytosis animation. If I was to click play, this would give me an overview of endocytosis and exocytosis.